So it's my pleasure to welcome you to this Keynote symposium, and a particular pleasure to introduce the first speaker today, and to welcome her to her first Biology of Genomes meeting. So because this is the first time she comes to the Biology of Genomes, it occurred to me that some of you may not know her work, even though it's had a tremendous impact in evolutionary genetics. And so I just want to spend 1 or 2 minutes introducing her. So, yes, so she's going to take a step back. I'm sorry. By way of introduction, she started her scientific career working with Bill Hamilton, father of kin selection, and then moved to Arizona, where she ended up spending many years as a faculty member at the University of Arizona, did a brief stint at Yale, and is now, for the past year, at the University of Texas, Austin. And she's the recipient of numerous awards early in her career, MacArthur Genius Award, more recently the International Prize for Biology. She's been a member of the National Academy for a decade, is in the Academy for Arts and Sciences. And what these prizes recognize is her pioneering work understanding the biology of symbiosis. And so for many decades now, she's been interested in elucidating an extraordinarily complex phenomenon that's fairly ubiquitous in the natural world, and that is the mutualist interactions that multicellular hosts have with microbes. And so in particular, she's focused on aphids and obligate and facultative bacterial symbionts. And she's used a dizzying array of tools from fields as varied as genetics and physiology and ecology to make sense of this system. And she's turned this entire system of non-model organisms into the best understood example of symbiosis. In fact, the textbook example that we use to illustrate basic concepts of symbiosis in introducing people to evolutionary biology. And so in particular of relevance to this audience and our motivation in bringing her here today, in part, is that she's used tools from what we would now think of as genomics and focused on microbiome decades before those became hot topics in human genetics in particular. And her work has demonstrated again and again the fundamental importance of microbial metabolic capabilities for host ecology and evolution. And so, without further ado.
Thanks a lot, Molly. That's quite an introduction. I'll try to at least somewhat live up to it, at least not go over. So thank you all. Let me get this set up here. Is that okay? So I'm referred to as a guest speaker. And I guess I'm a guest since it's my first time at this meeting, and I've really enjoyed it. Fantastic, outstanding talks, and I've learned a lot. I'm also going to be talking about guests, the sort of guests that live in the bodies of animals. And in particular, I'll talk a lot about insects, the systems I've worked on. But there are many parallels in a lot of other invertebrate groups. Vertebrates tend to be a bit different and not have these kinds of symbionts particularly, although we may still discover some. But this is a very widespread phenomenon in many different animals. Okay, so this phenomenon referred to as the resident genome phenomenon, it's really, it's basically that a host-restricted or symbiotic lifestyle where organisms live only in a host and don't replicate outside of it leads to genome reduction and accelerated sequence evolution throughout the genome. And this has turned out to be a ubiquitous pattern of convergence in genome evolution. It's across the tree of life. It's an irreversible evolutionary path, so lineages that go down this route don't seem to ever go back out of it. And we see cases, particularly in bacteria. Many different phyla of bacteria have produced these kinds of lineages, but even in archaea and eukarya. And the extreme cases, it turns out, are in insect endosymbionts, possibly because we've studied them a lot. We may find equally extreme cases in other, for example, marine invertebrates. But even within insects, there are many repeated examples of it that's evolved again and again. So there's a lot of convergent evolution in these lineages. So I'll just start with kind of an example. We've studied a bunch of cases, and today I'm going to be summarizing kind of the overview that we have of this from a bunch of different cases. But I'll use our favorite recent example. Molly mentioned aphids and Buchnera, which is kind of the classic model. But there's some other insects that also have very interesting and sometimes more complex situations. This is the case of these leaf hoppers. So here's your insect here. It has some paired organs here in the abdomen, which are called the bacteriomes. They're composed of a bunch of cells that house, in the cytosol, they house bacteria. And so here's a picture of one of those, and this is some in situ for the ribosomal RNA of the symbionts. It turns out in this insect and in all related insects, there are two symbionts from different phyla of bacteria. One of them is called Solsia, one of them is called Nesuia, and they live in these distinct cells within this organ. So here's a bunch of cells. The blue dots are the insect nuclei stained with DAPI. These symbionts are typically in all these systems maternally transmitted, so that's a key aspect of their evolution and probably why they are so extreme. Here's a picture of an egg, the egg mass, and the symbionts are deposited as a mass within the mother's body cavity. The symbionts are inoculated into the eggs and transmitted. And it turns out that doing lots of phylogenetics on many of these different groups, this is really the way I started working on these systems, was doing phylogenetics of the bacteria and the insects 20 years ago. And it turns out that again and again we see this pattern of matching phylogenies between the insect and the symbiont, and in cases like this, between the two symbionts. So basically you get these parallel phylogenies which result from very long-term maternal vertical transmission of symbionts. In case, for example, this is one of the more ancient cases, 270 million years, the very base of a very large group of insects that includes cicadas and leafhoppers and others. So we have this very long-term evolution within the host. So why are they in there? Just a quick note on that. It turns out there was someone who studied these early on with microscopy, Paul Buchner, a German biologist, and he hypothesized that the role of these kinds of symbioses, he was just using light microscopy, he really had very, very limited ability to look at things, but he hypothesized that their role was in nutrition, and that has largely turned out to be the case, that these symbionts are providing essential nutrients. And in the case of these insects, they feed on plant sap, either phloem sap or xylem sap. And when you look at the contents of plant sap, it's very biased, particularly in amino acid composition. So essential amino acids, that is the ones that animals in general require, including us and including insects. So basically, those pathways were lost early in the evolution of metazoa. Essential amino acids are very low in plant sap. Well, those are the expensive amino acids. They're more expensive to make, and they're not used by plants in general as a way of moving nitrogen around. And so an insect feeding on plant sap has the problem of how do they get enough of these, and the symbionts are making them. And the genomics has really confirmed that. So what we do is we sequence symbiont genomes. It's become easier and easier over the years. It used to be expensive and hard, and starting in 2006, 2007, it got cheap and easy, so there's more and more of them sequenced. These genomes, even though they're small, which is the main thing I'll be talking about, they retain pathways that are the exact ones needed by the host to produce the limiting nutrients. So here's just one example. You can reconstruct what pathways are encoded in the genome. You sequence the genome, you have very clear homology, and a lot of these pathways, there's only one way to make tryptophan, only one biosynthetic pathway. It's easy to recognize the genes, and they'll have all 7 genes, for example. So you can count up what they can do and score the metabolic capabilities of different symbionts, and you get these beautiful patterns. This is really mostly the work of John McCutcheon, who was a postdoc in the lab. He's now at University of Montana as an assistant professor, but he was a postdoc. He came right when next-gen sequencing started in 2006 and 2007, and it was the perfect timing because he was very good at it and very good at the informatics for putting these genomes together. And he sequenced genomes from several insects that had these paired symbionts. So this one called a sharpshooter feeds on xylem sap. So it has one symbiont that makes these 8 essential amino acids. The other symbiont, which is a completely different thing, the gamma proteobacteria, makes the other 2 amino acids, histidine and methionine. Then he did the cicadas, the big insects that sing in the trees. So it has, again, the Solsia symbiont, and it has a completely different partner, an alpha proteobacterium, which again makes histidine and methionine and no other. So they're perfectly complementary, and it has this bad luck of making methionine using the alternative pathway, which is dependent on cobalamin, which is a very complicated molecule. It takes many genes to make, so this genome also makes cobalamin, probably just for the production of methionine. And then finally, in these insects, the spittlebugs, the Solsia makes 7 of the amino acids, and its partner, which is a beta proteobacterium, makes the 3. It actually adds the tryptophan. So you get this really reassuring patterns that genomics really is telling you something about the metabolic capabilities in these relatively simple sort of metagenome systems. This is just a list of some of the main ones. The list has gotten too long to put on a slide, but these are just some of the insects that have been studied, many of them by us, but also some other groups. So aphids, carpenter ants, the little ants that are walking out there on the wooden benches when you want to sit down, tsetse flies, the vectors of some disease, sharpshooters, psyllids, cicadas, cockroaches, mealybugs, spittlebugs, whiteflies, and others. So there's all these different insect groups that have independent cases of symbionts that have invaded them. Most of them are gamma, beta, or alpha proteobacteria, so proteobacteria, or Bacteroidetes, okay, which are phyla that are commonly associated with animals. They're often in the guts of mammals, for example. And they have these very tiny genomes. So the first genome sequence was in the year 2000. It was done in Japan, and that was a Buchnera and P aphid. And it was a very small genome. The main result was this is a very small genome, fairly close to E. coli. It had about 1/7 of the genes of E. coli. So it was a subset of the E. coli genome, essentially. But as time went on, and especially there was a transition in 2006, we realized these can actually get a lot smaller. So they're not just half a megabase, they're only 0.1 megabase. It's gotten down to it. And so there was kind of this realization there seems to be sort of no obvious limit to how small they can get. Well, in bacteria, their genomes are very simple, which is one reason I study them. They're elegant and simple. Most of the genome is doing something. They're pretty much 85 to 90% coding sequence in bacterial genomes, and even the parts between genes are often doing something. So you get this beautiful pattern. The genome size is very strongly correlated with the numbers of genes in the genome. And now and then you get a few dots falling down here, which are basically organisms that have a buildup of pseudogenes, genes that have been inactivated. But we're interested in these ones down here at the bottom, these obligate symbionts of insects, which include all of the extreme. I think the 6 smallest bacterial genomes are insect symbionts. The smallest one right now, as of this year, is Nasuia in leafhoppers, 137 genes, 120 kilobases. But you see that the pattern actually starts, even with host-restricted pathogens, they actually show many of the same patterns, just not as extreme. And here's just another picture. Here's Mycoplasma genitalium, which you might know from Craig Venter. It's considered kind of a minimal genome. It is the smallest. It is the organism with the smallest genome that can be grown in a pure culture in the lab. And here are just the relative sizes of some of these other ones, just to give you a picture of that. So why are they so small? What is the basis of this resident genome syndrome? Well, first of all, imagine a bacterium becomes a symbiont, a free-living bacterium becomes symbiotic. Things are suddenly very different. It doesn't need to do a lot of the things it used to do. It can get a lot of stuff from the host. It doesn't need to deal with a lot of substrates that it used to encounter. So many genes are not needed. There's relaxed selection, and they can be eliminated. And we know in general that in bacteria, genes that are not needed are eliminated. That's why their genomes are consistently streamlined. Bacteria have lots of genes coming and going in your normal bacterium lineage. There's lots of genes coming in, lots of genes going out, but you don't see a buildup of sort of dead genes, of junk DNA of any kind. Basically, they're very streamlined. There's a lot of deletion, so basically, to keep DNA in a bacterial genome, selection needs to be maintaining it, and that is a pattern that really is true in bacteria broadly, and something of a contrast with eukaryotes, with most eukaryotes. But this relaxed selection is really not enough to explain what you see, because you lose genes in all categories, even ones that are involved in the just machinery of the cell, translation, transcription, sort of accessory factors of all kinds. Basically, every functional category, you lose genes. So it's not just that there's a change in ecology, because there's basically all kinds of genes are lost. For example, DNA repair genes there are always many of them lost. Sometimes essentially all of them are lost. And the other part of the explanation is this change in population structure, that these host-restricted bacteria have much smaller effective population sizes, and they are much more clonal than free-living bacteria. So it takes more selection to maintain something. So a gene that's only weakly beneficial, that could be maintained through a weak selective advantage in a large free-living bacterial population, won't be maintained when you have this switch to a completely different population structure with a smaller population size, because it's basically based on the population size of the host, and the animal host will have a smaller population size than free-living bacteria. And then a major thing is that they're clonal. right? Especially these maternally transmitted one, they're strictly clonal. They have the same population genetics really as mitochondria or plastids. Okay, so you get increased levels of genetic drift, fixation of deleterious mutations. Some of these are basically inactivation events that kill weakly beneficial genes, and then that DNA is deleted, and this results in genome reduction. Some of these mutations will simply be result in the change of amino acid that makes the protein less stable, maybe less efficient, less stable, and there'll be a buildup of those too, because those also will get fixed more often. Okay, well, some of the genes that are deleted are DNA repair genes, and so this could actually accelerate this process further because the basic mutation rate will be elevated because DNA repair basically reduces mutation rate, and when you remove those in bacteria, knock them out, you get massively increased mutation rates. Okay, well, I'm asserting all of this, and basically there have been a lot of different studies done starting back in the middle 90s, and many different, basically every time a new symbiont group is discovered and studied, people do more studies showing these kinds of phenomena. Here are the things that make it look like genetic drift is the explanation for this syndrome. There's high Ka/Ks genome-wide, very fast protein evolution relative to DNA evolution, lower thermal stability of proteins, and basically to be functional, you have to if there have been experiments putting them in E. coli, you have to overexpress chaperonin, the GRO-EL protein, to get them to function at all, to fold correctly. So basically they're dependent on high levels of chaperonin. There's random use of codons with respect to GC content, so this is an indicator that weak selection is not very effective in these. In most bacteria, there's adaptive codon bias in many genes. Within populations, there's low polymorphism and an excess of rare alleles, so the patterns you'd expect if you're having slightly deleterious mutations being fixed. And the absolute mutation rate, in Buchnera we have one pretty good measure of the absolute mutation rate per generation per unit time, and it is elevated over what it is in other bacteria in that group. So here are some of the references. It's based on just a bunch of studies. There's this sort of syndrome, and a lot of things fit with it. Here's a tree of bacteria. So this is, almost the tree of life. It's the tree of all bacteria. Here's the alpha proteobacteria, gamma proteobacteria, the Bacteroidetes over here. And these long branches here are these insect symbionts. So this is a protein-based tree, and they have massively accelerated rates of protein evolution. You can still annotate the proteins, and particularly annotation obviously can be sometimes complicated when you have really crazy evolution like this, but the active sites or the conserved sites of the enzymes and so on will be conserved. You'll be able to find them, and basically you can make a tree. It's just you get these kind of insanely increased rates of evolution. So most of these small genomes are also very biased towards A and T, genome-wide. There's a couple of exceptions, but most of them are very AT biased. The most extreme are something like 13 or 14% GC, which that's mostly protein coding genes. So just imagine what that does to the protein, and it has a massive effect on the amino acid composition of all the polypeptides encoded in the genome. And to me, this is a very strong argument that this has got to be deleterious. For example, if you take alanine, if you take an AT-rich genome, it'll have tenfold lower frequency of alanine because alanine has to have a G and a C in the codon family, but it'll have a 10x higher frequency of isoleucine, which has an A and a T. So basically there's this sort of mutational pressure towards A and T that's having a massive impact on polypeptides. Okay, well, we have a bunch of these independent cases. Here's all these tiny genomes, different ones and from different insects. We also have a number of cases where there's a obligate endosymbiont that's actually closely related to a free-living bacterium. And those are interesting to kind of get an idea of what are the early steps in this process of genome reduction. And so here's a few cases where if we look at the 16S genes, here's a Serratia symbiotica, the 16S is very, very close, just a couple percent different from free-living Serratia, suggesting that it's a rather recent divergence, and that's the same for a couple of other cases. And so we can look at these genomes and say, what does it look like in the early stages before you have this extreme reduction? And this is just showing a couple of them. So here's sort of normal Serratia proteomaculans out here, and Serratia symbiotica. You have this massive decrease in genome size, actually an even greater decrease in the number of intact genes. That this y-axis is the number of genes that are still intact. And this is because it has actually built up a bunch of pseudogenes. And so basically you get a change from sort of a normal genome with few pseudogenes packed with genes to these much reduced genomes that have a buildup of a lot of pseudogenes, so that's why they're falling down below this line here, and massive reduction. And that early reduction, part of it has to occur through large deletions. If you just sort of calculate the rate of DNA loss, like how you could get from here to here over how many million years could possibly have been in there, some of the DNA loss has to be through large deletions, many genes deleted at once to get down that small. Here's a comparison of the Serratia symbiotica to two free-living Serratia, they're all about the same distance apart in terms of just sequence divergence of orthologous genes, but this symbiotic one has a massive number of genome rearrangements. It's got a lot of insertion sequences, transposable elements, and repetitive sequence, lots of rearrangements, and it clearly has had many large deletions, possibly mediated by recombination between these insertion sequences, which take off and explode in these kinds of genomes. And if you look at what genes are being eroded, basically they're in all different functional categories. So these are the different cog functional categories for different kinds of activities. Basically all of them, the Serratia symbiotica is this middle blue one here. Basically there's pseudogenes occurring in all of these, loss of genes in all of these. And one thing this tells you is this is not driven by selection to have less DNA in the chromosome. In many cases, the gene dies just through an inactivation event, a stop codon or something, a frame shift, and then is slowly eroded, right? So often there's not an immediate loss of DNA. So it seems there's just not enough selection to maintain the function of these. So what about these tiniest genomes? Are they at a limit? How small can they get? Do they just stop? What happens at that point? So these poor insects are dependent on these kind of vestigial little organisms that are still pumping out nutrients that the insects are dependent on. So is there some kind of limit? Well, the first one of these tiny genomes we found was this one called Carcinella ruddii. It's in a group of insects called psyllids. They're a lot like aphids. They feed on phloem sap. And it had a genome size of only 160 kb. And actually, we had discovered this at least 6 years earlier. I think 8 years earlier, we'd done some pulse field gels, and we kept getting this 160 kb fragment. And we just didn't believe that was a whole genome, so we just assumed there was some kind of weird artifact. It turned out it got fully sequenced in collaboration with a group in Japan who did the sequencing. And the lead author was Atsushi Nakabachi on the initial genome, but then Dan Sloan more recently resequenced a bunch of different genomes and tried to ask the question, when you have a really tiny genome like this, is it still losing genes, or did it sort of reach a limit and is just staying static? And it turns out that whenever you do one of these studies, as in this case, there's still some gene loss occurring even among these related insects. So basically there doesn't seem to be a kind of absolute limit to, genes are still being lost over time, even when you do sort of closer comparisons. So it doesn't seem as though it necessarily stops. Another feature in general in these comparisons, when you look at more than one genome within one of these clusters, is that you see no gene uptake at all. So over whatever period of time, they're not taking up genes. Most bacterial lineages are acquiring genes through horizontal gene transfer at some rate. These are not. And there's no gene rearrangement, essentially no gene rearrangements, in this case none. In almost all cases there's actually none. So even in comparisons for Buchnera and other symbiont groups, we see again and again this pattern. So these are very static genomes. All they can do is lose genes. So what is missing? Well, some people immediately say these are just organelles, and it's fine if you want to call them that. I'm happy about, you can call them that. But they're quite different from mitochondrial and plastid genomes. They're much more cell-like than those. They mostly retain genes for replication, transcription, and translation, at least the core genes. For example, ribosomal proteins pretty much are contained. So these are the different symbiont genomes represented from the sort of the larger ones out here. So here's Dalmania and Buchnera's here, and then here's the tiniest ones in here in terms of overall size. And you can see they basically retain the ribosomal proteins. They retain some chaperones. They retain the core genes for replication and transcription. So RNA polymerase, DNA polymerase, they always keep that. But they keep a pretty good set of tRNA synthetases, one for every amino acid, although there's a couple that will be lost repeatedly that are just mysterious, like how is this working without those? Clearly there's some trick, and it's happened more than once, and we don't know exactly what it is. But in general, they're kind of keeping like what you would think are the set of genes. If you asked an introductory, cell biology or molecular biology student, list the genes a cell must have in order to replicate itself, it mostly keeps those genes. There's a couple of really interesting things. For example, we found 2 independent cases of a recoding of the UGA stop codon, and they're associated with loss of this gene for release factor 2, which the protein is the release factor that recognizes this stop codon. And what we think happened, so initially this became apparent just from doing alignments and observing that there were stop codons every time where there should be tryptophans as conserved across a whole bunch of other bacteria. So we reasoned that this must be a recoding, and then John McCutcheon did the proteomics to prove it was a recoding, and we found 2 different independent cases. And probably this results from initially the loss of release factor 2, this PRFB. That's deleterious, right? I mean, losing the release factor that recognizes one of the stop codons is a bad thing because the proteins will be too long, but it could be functional. I mean, they're already barely functioning. They're within the host, they're sort of protected. Of course, then there'll be selection immediately to replace the UGA stop codons with UAA stop codons. And then that frees up UGA to encode tryptophan. And so there's only one other case in mycoplasma where this has happened in bacteria. It also has a small genome. Where you really see a lot of stuff missing is in the kind of energy metabolism. And here it seems like every organism, every one of these has a different solution to what they do. Some of them have more or less complete sort of energy capabilities. Some of them have lost NADH dehydrogenase. Some seem to be total ATP parasites. They can't do anything in the way of energy metabolism. And so there's a whole range of what they do, with the smaller ones being the most host-dependent in general. Some of the gene categories where we really can understand why you could lose a lot of genes maybe is those, basically all the genes, which is a lot of genes involved in production of the cell envelope, peptidoglycan, phospholipids, and cell architecture. In a normal bacterium, there's a large set of essential genes for these things. These are very, very reduced in these organisms. And this may be an arena where the host actually can compensate. The host can provide components, transporters and membrane components that can gradually, over millions of years, substitute for the cell envelope of the organism. And that seems to be what has happened. In some of these, basically they can't make any of their own cell envelope. It must be coming from the host. And I think I forgot to mention that the typical arrangements, they're in the cytoplasm of the host, but they are surrounded by a host-derived membrane. They're in a vacuole within the host. That is the general arrangement. So that membrane could evolve, since the host is being selected to support these symbionts, could basically start taking the place of the symbiont's own cell envelope. Transporters, again, these are essentially all missing, even though we know amino acids go in and out. There's experimental proof of it. These are all more or less missing, and probably this is another arena where the host has taken over. And then regulatory genes, the classic bacterial regulatory genes, both the global ones, the specific ones for pathways, are essentially all eliminated. Just a handful will be retained, like Buchnera maybe has 5 instead of 50 in E. coli. And again, this is probably something else is going on. There's a couple of just very recent studies that are in the works that give some suggestion about host-derived molecules that could play a role in sort of regulating the cell cycle of the bacteria. But that's kind of one of the mysterious areas. So how do they keep functioning? Well, obviously there could be some counteract, some compensation. And one thing we know that happens in the bacteria themselves is all of these systems, they overproduce heat shock proteins constitutively. So that in Buchnera, 10% of the protein in the cell is this GRO-EL protein, and basically this has been true again and again in these small genome organisms. They overproduce heat shock proteins, which sort of keeps the interpretation is that this, and there's some experimental evidence, basically high levels of chaperonin can enable their enzymes and so on to continue to function. And the other side of the coin, though, in these mutualistic symbionts is that the host is being selected to take care of them, right? And this is probably the general reason that these mutualists can evolve smaller genomes than pathogens, right? That hosts do not evolve to take care of their pathogens, but they do evolve to take care of the symbionts upon which they are dependent. And so this is the reason, presumably, that they can become much smaller and lose more genes. And there have been several studies now on these three systems, aphids, mealybugs, and psyllids, done by our lab and also John McCutcheon's lab, looking at gene expression of the host in the bacteriocytes. The host cells, those very specialized cells that house these bacteria, have distinctive patterns of gene expression. Here's just a picture of a bacteriocyte in an aphid. It's a large cell. It has about 10,000 Buchnera in it, which are these sort of round bacteria here. Each of them is enclosed in a host-derived membrane. Here's the single nucleus in the bacteriocyte cell. And then, so this membrane around the Buchnera cell could be very important in basically gating the sort of metabolism. Allison Hansen was a postdoc in the lab. She's now at University of Illinois. She did a study of gene expression in bacteriocytes in aphids, which involved picking out by hand thousands of bacteriocytes out of aphids. They're large cells, but still it's pretty tedious. And then doing an RNA-seq study. And she took advantage of the fact that the P-aphid genome had been sequenced. And she found a really striking pattern of high expression in the bacteriocyte specifically of a set of genes involved in amino acid metabolism. So that basically there are little amino acid factories. And so on the aphid side, it has upregulated many genes involved in amino acid metabolism that complement the capabilities of the bacterium. So recycling ammonia, aspartate glutamate interconversion. Amino acid transporters are very highly overexpressed in bacteriocytes. Actually, one of the steps in the final pathway, the final step in the branched chain amino acid pathway is encoded in the aphid genome, not in the Buchnera genome, and so on. So there seems to be this real complementary aspect. Then finally, another thing that's become really interesting recently is the question of are bacterial genes transferred from the symbiont to the host genome? So we know this has happened in mitochondria and in plastids. Many genes from the ancestral symbiont have gone into host genomes. That was in most of that occurred when the host was a single-celled state, though. What about insect symbionts? These are mostly occurring in somatic cells. They're only briefly in the germline cells. Has it happened? And we now have 3 cases where we can actually look at it because there's been enough sequencing of the host genome. So the aphid Buchnera was the first one. The P-aphid genome was sequenced. It turned out there were 11 genes, very clear-cut, all checked out, that were from bacteria that are part of the aphid genome. However, they weren't from Buchnera. So they came from other bacteria, and most of them seem to come from Wolbachia, another symbiont that's commonly in insects, although it's not in P aphids now, but was probably in an ancestor. So an ancestral symbiont probably gave it. There's also mealybugs and Tremblea. This is the most striking case. I'll mention it again in a minute. It had a whole collection of genes from bacteria, including Wolbachia and some other symbionts, but none from its main symbiont, which is called Tremblea. And then psyllids and Carcinella, a similar pattern. It had a handful of genes from bacteria, including Wolbachia. It only had one from its main symbiont, Carcinella. So these host genomes have bacterial genes, but they're not from the symbiont. They're from probably past symbionts. But when we look at these transferred genes, they actually seem to play a major role in the bacteriocytes. They're upregulated in the bacteriocytes. They seem to play a role in regulating the bacteria. Some of them are lysozymes, some of them are involved in other aspects like phospholipid and even peptidoglycan production in the insect genome is overexpressed in the bacteriocyte, suggesting that indeed the host is contributing to the cell envelope of the symbiont. And we've seen in each of these cases some instances of actual pathways where you can look at the pathway and there'll be some steps that are carried out by a gene from the symbiont, some that are from a host gene of eukaryotic origin, just an insect gene, and some a host gene of bacterial origin. So this is a case in aromatic amino acid production in psyllids. But the most dramatic case is one that John McCutcheon studied. So this is actually from his lab, and it's on mealybugs. So this is kind of the most extreme kind of case of the fusing of genomes. So here's a cell, a bacteriocyte. In it, there's another bacterium here. This is Tremblea. So here's a the nucleus, the host nucleus. Here's a Tremblea, here's another one. And then in the Tremblea are some other bacteria. As far as I know, it's the only case I know of where bacteria live in another bacterium. It's a very rare thing. And this one, well, he named it Moranella, so it's living inside a symbiont, inside these cells in mealybugs. And it turns out that there's this whole kind of mosaic of sort of gene exchange that's occurred so that the host genome has a collection of bacterial genes. There's a number of pathways for different metabolites, essential amino acids, vitamins, but also even like production of peptidoglycan that seem to be complete, but only if you include genes from each of these different sources. Right? So it's actually quite an extreme and bizarre case where it really looks like you're sort of putting together sort of steps from several different genomes to produce basically essential functions for the overall organism. Okay, well, just to summarize, there's this syndrome, the resident genome syndrome. It's associated with massive loss of genes, and you get these tiniest known genomes. And it's a case of convergence without adaptation, right? I mean, at least I'm largely arguing that this is due to fixation of deleterious mutations, loss of genes that actually would be useful, but there's not enough selection to maintain them. Of course, there's some adaptation. I mean, I'm not saying there's none, but the overall syndrome seems to result mostly from genetic drift. These genomes also show other very extreme features, very fast sequence evolution, codon reassignments, extreme AT bias, things that are quite rare. Are they organelles? Like I said, it's okay with me if you call them that, but they're more cell-like in general, and they make their own ribosomes, which I count as a separate organism if it makes its own ribosome. The tiniest genomes are all in these mutualistic symbionts, mostly lineages of proteobacteria and Bacteroidetes. Those are phyla that commonly associate with animals, so they're the ones that have moved into these situations. And hosts evolved to support the symbionts, and this probably just leads to even more erosion, right? You support them, and then they erode even further. Since 2010, we've been seeing more and more cases of animal genomes that have incorporated genes from bacteria and also from fungi. And these often seem to be from ancestral symbionts, so it's sort of another major role of symbiosis in animal evolution, is actually getting genes from them. And I think there'll be more of these cases because it seems like we see the first case, it seems like a really weird one, but then suddenly there's a whole bunch more cases. And it's not going to be a large proportion of genes in these animal genomes, these invertebrate genomes, but they may be ecologically very important or kind of critical for like maintaining a particular ecology. So we'll have to see how much that turns out to be true, how common they end up being. So just to acknowledge, I talked about John McCutcheon's work. He came in. Ryu does a lot of the microscopy, the beautiful fish images, and also Gordon worked on the leafhoppers. Allison did the RNA-seq with the aphid bacteriocytes, and Dan Sloan worked on Carcinella primarily. And with that, and the funding's NSF mostly. With that, I will stop and hope I left time for questions.
Questions?
Amazing talk. So my question is, like you said, the endosymbionts can divide within the bacteriocytes that house them. Do you think that's actively regulated by the host? And how does that work when you have multiple types of endosymbionts, like primary and secondary, within one host cell?
Right. So typically, there are not more than one type of symbiont within one host cell. And I think that you sort of hit on it, basically is not stable. So when there's two symbionts, they're usually in two types of bacteriocytes. And we've seen cases, I didn't go into all the details, but we've seen cases where an ancestral symbiont has actually sort of been replaced by a new symbiont. You know, it might be a way of getting a better symbiont, right? But when that happens, it doesn't simply invade the ancestral bacteriocyte type. A new cell type evolves that houses the new symbiont, right? So, and then the first part of your question, yeah, there's still some mysteries here. There's got to be regulation of the cell division of the symbionts by the host, or some kind of feedback, right, so that they don't over-replicate and so on. And the nature of that's not really known. So experimentally, these can be very difficult to work on. You know, obviously we can't grow them. I didn't even mention that, but we can't grow them outside of the host. You can do a lot of things, though, and sometimes you sort of get lucky, and I think we'll start to see some progress on that. I know of a couple people working on things that look really promising for how the host might control a symbiont.
Hi. So, kind of a related question. There's selection over the generations of the insect for the symbionts to produce these essential amino acids. But throughout the lifetime of the individual insect, what stops the symbionts losing that and sort of breeding without having to make them? So, what selection pressures keep the symbiotes from being symbiotic during the life cycle of the insect?
Right. Well, there is a potential sort of for cancerous like symbionts, right? I mean, and although basically selection at the host level, I mean, it's sort of a levels of selection thing. Within host selection could favor selfish symbionts that over-replicate and are actually detrimental or that don't make tryptophan or whatever the host needs. But the limitation on that would be selection at the host level, right? Those hosts won't reproduce, and the only way these symbionts persist is by being passed on maternally from their host. So, you know, there is a conflict, but you know, symbionts that don't allow their host to reproduce will not be around the next generation, right?
Wonderful presentation. I have a question about the recoding events. So there are many possibilities as to what could be leading to that. Number one is that they're losing tRNAs, and therefore, you know, they have a reduced code, and therefore they end up sort of recoding their genes for that. Another is perhaps as a way to avoid transfer of genes between the host and the parasite. Sorry. So my question is if mom is calling
Maybe they
again.
know the answer, yeah.
So my question is how frequent are these events? Is it more than what you would expect?
Yeah, they're not. Well, we found two, and there's one other in bacteria, and there's quite a few in mitochondrial genomes in different weird mitochondrial lineages. And this particular reassignment is the most common one. And I think, you know, we did some calculations, like that codon was still in the genome when, I mean, even if they're AT biased, you don't go down to, there's still a handful of codons of that type that would be used. So somehow it happened. I mean, there's no easy way for that to happen, right? No matter what, there's some kind of jump. But I just see it as, rather than some kind of really nuanced adaptation to avoid gene transfer, why would it care if its genes are transferred to the host? I mean, you know, that's kind of to me, it just seems like another case of gene loss, and it somehow survived. And we see a lot of others that are a little hard to explain too. And one of the cases happens to be, one of the explanations has to do with very strong base compositional bias towards A and T. But it happens that one of our two cases is one of the GC-rich ones, and one of them is very AT-rich, one of them is GC-rich. So it doesn't seem like it's base composition driving it.
Translation inefficiency, basically.
Right.
Wow.
Yeah.
Fascinating. Thank you.
Any other questions? Yeah, one more question.
Okay. So there's increasing evidence that insects are basically derived crustaceans, with their closest living crustacean relatives being cephalocarids and these remipedes. And there's some knowledge that remipedes have endosymbionts too. And you said that there's evidence for ancient symbioses that no longer exist, genes transferred from symbionts that we don't see anymore. How far back does bacterial symbiosis go in the insect tree?
Yeah, well, I think it probably goes back before there were insects. I mean, I think it goes all the way back. I mean, every insect group, these lineages from the very base of insects can have symbionts. I mean, there's particular ones we focus on where we can pretty much date when this one came in, because this whole clade has it and the things outside of it don't have it, right? So that big clade that's 270 million years, it has cicadas, spittlebugs, leafhoppers, it uniquely has these symbionts and other things don't. But probably, even at that stage, there were some symbionts in those ants. There's no reason to think they would have been absent in the past when they're ubiquitous now.
And their sister group has them.
Right, exactly, yeah. And actually, I should mention other invertebrate, like marine invertebrates, it's being found more and more that vertically transmitted symbionts in sponges, corals, so on, that occur. So it's probably quite widespread. Insects are just what we studied and what Paul Buchner, this German person, studied. He wasn't near the sea, so but there's no reason to think that it's an insect-specific thing necessarily.
Wow.
Okay, one last question.
Thank you. So you presented that low effective population size, increase stochastic drift and accumulation of mutations, deleterious deletions. And if there is no limit in this process, so it seems likely that the final step in this story is just extinction of symbionts. What do you think?
Well, I actually think that's possible. That the insects, are dependent on these, extremely inefficient kind of. A lot of them have, one thing I didn't go into. They have very limited temperature range, for example, which there's basically Buchnera can be very, very temperature sensitive. So they can limit the range of their host. Actually, Buchnera limits the temperature range of aphids. The Buchnera will die, and the aphid can't reproduce, so it dies too. So, it does seem possible that having such dependence on, such a kind of an eroded genome could lead to extinction of some lineages. It's a very hard thing to, test, obviously, because we don't see extinct things. So,
Thanks again.